It's my great pleasure to introduce our next speaker, Lorena Beese. So Lorena is the James Duke Professor of Biochemistry at Duke and a member of National Academy of Science. So Lorena is actually a prime example of Tom's legacy. So Lorena did a postdoc as a postdoc fellow in Tom's lab in the late 80s and the beginning 90s. And she continued to work on DNA repair and DNA replication, and now has become a foremost leader in the field. So Lorena is going to tell us how this was all started with working with Tom.
That's how he was in the beginning. Yeah.
I can, whatever you think is going to work, yeah. Great. Well, it is a real honor to be here today to celebrate Tom Steitz and to present some of his seminal contributions to understanding DNA replication and DNA polymerases. I mean, it seems like yesterday that former lab members, students were all gathered not too far from here to celebrate his Nobel Prize and to entertain Tom with stories of research that we had done since leaving his lab at Duke. But today, rather than tell you some of the stories I've done since leaving, I thought I would share with you some of the excitement for working with Tom as a postdoc back in the late 1980s and early 1990s, just as he was starting to put together the first molecular pieces of the central dogma, specifically how these DNA polymerases copy DNA and do so with such high fidelity. Well, for the students in the room, I just want to say that accurate DNA replication is essential not just for genome stability, but and defects of the process also underlie so many human cancers. And broadly speaking, we know now that there are basically 3 stages to the process where the high fidelity polymerases enhance the reaction, making errors in every 1 in 10 to the 5th, 1 in 10 to the 6th nucleotide synthesized. And this takes place at both the insertion step and at the extension step of the reaction. And then if a mismatch does get incorporated, it causes these enzymes to stall, and then the mismatch can be excised by these proofreading exonucleases that further enhance the fidelity of the reaction several orders of magnitude. And then lastly, if it escapes the replication polymerases, we have the strand-directed mismatch repair system to go take out the bases and further enhance the process. And so my passion for understanding these processes began when I was a postdoc in Tom's lab, continues to this day, although we're mostly working in this part of the problem at this point. So when I first joined Tom's lab, he had just solved the structure of the first DNA polymerase, that of DNA Pol1 from E. coli, shown here, and this was published in 1985. It was actually the first polymerase ever identified back by Arthur Kornberg several decades earlier. And the structure was a real breakthrough at the time, being the first polymerase, because it provided a framework for trying to really understand these mechanistic questions. It has a polymerase domain coded here in color-coded in yellow, and a exonuclease domain colored here in blue. And one of the striking things you see is the absence of DNA. There's a big gap in here, and all that we have marking the active site is the nucleoside monophosphate, which is the product of the exonucleolytic reaction, pointing out where the exonuclease site is. And the fundamental questions at this time when I joined were really fundamental and basic, like, where is the polymerase active site? And Tom would joke, the crystal structures do not come with roadmaps. You have to figure out where these things are. And if you can't see them directly with your crystal structures, then you have to rely on biochemistry and genetics to try to figure it out. And so, fortunately, at the same time, Tom was beginning his collaboration with Nigel Grinley and Kathy Joyce over in the medical school, making mutations in this area to try to figure out where the active site was. And indeed, the residues that are color-coded in red were absolutely essential for catalysis, and the tyrosine marked in green was the site of cross-linking of a triphosphate, so clearly the active site needed to be someplace here. And then additionally, Kathy was going and footprinting the DNA and trying to figure out how much was being protected by the polymerase, and it was about 8 base pairs, if I remember. And this led to Tom making a model for how the DNA would come into the active site right to where the triphosphate was, identified by cross-linking. That would be the primer terminus. The template would be coming through like this. Down here, the exonuclease. It was such a beautiful model, and many of you may have first seen this model when you were taking biochemistry, because it was in so many of the biochemistry textbooks at the time. And it was an incredibly useful model for thinking about the problems of, well, how do these active sites work together? My goodness, they're 35 angstroms apart. However, the polymerase was a molecule that had many puzzles, and this was going to be one that was going to be revised with a new model as more data came in, but it was incredibly useful at the time. So actually getting complexes to prove this model was really my primary task as a postdoc. And try as I might, that was not going to happen, or didn't actually happen. But instead, the exonuclease was much more tractable, and we readily got complexes with DNA substrates in the exonuclease active site. And that, combined with the monophosphate product, allowed us to begin to think about mechanism. But it really wasn't intuitive. Here is the reaction that's carrying out in the exonuclease active site. You have the enzyme, this is the primer and the template, and you're going to be clipping off, the enzyme's going to be clipping off this nucleoside monophosphate product from the primer strand. And naturally, you would be thinking that this should be taken, as this figure might suggest, it should be taking place at the site of synthesis. But it doesn't. It's 35 angstroms away. And so, how does this actually happen? Well, one thing that was clear from early biochemistry studies is that it was actually a single-strand DNA that gets into the active site. That in and of itself was puzzling. However, the crystallography made that part of the puzzle clear early on. So here we have an image. It's not looking too good, but we have the space-filling representation of the Klenau fragment with a 4-nucleotide substrate bound, and the base you can just see peeking out there is the nucleotide that's going to get cleaved, and the rest are extending up toward the polymerase active site here. And then if we sort of zoom into this area, this is where all the action was, both for catalysis and in terms of all of our thinking at the time. So here we see an image of the substrate coming into the exonuclease active site, surrounded by key residues and two critical metal ions. And so to try to figure out how it worked, Kathy and her students over in the medical school were systematically mutating all the residues within a very large radius of the nucleoside monophosphate and subsequently of the single-strand substrate to try to figure out which ones were important. Meanwhile, we were crystallizing many of these mutants to try to see if we could understand what they were doing based on the structure. And to summarize an awful lot of data, it turned out that really it was only the side chains that were coordinating these two metal ions that were absolutely essential. Everything else had ancillary roles, but these metals were really critical. And so in two back-to-back papers in EMBO in 1991, we published the mechanism for the 3 to 5 prime exonuclease, the two metal ion mechanism. The other key thing in puzzling this through that Tom was just so pleased about is that he didn't need to invoke any big conformational changes of the protein in order to get this mechanism to work. And that is that the nucleoside monophosphate product and the substrate almost completely superimposed, minus a slight rotation here at the scissile phosphate. And so the mechanism that he published was the two metal ion mechanism for DNA cleavage. And metal ion 1 would be activating a water molecule. It would go, he relied on the kinetic data of Gupta and Bevankovic, which said the reaction would go with inversion of configuration, so it'd be an inline attack. Another metal ion would be stabilizing the pentacoordinate intermediate and facilitating the leaving of the single strand. Now, I have to say that as we were working on this, Tom immediately recognized the generality of this two metal ion mechanism and would often say with a twinkle in his eye that it did not escape our attention that this could be quite general. And this is because it did not require any specific side chains. All that was required is that you had the metals in the right geometry to straddle the phosphate that was the phosphodiester bond that was going to get cleaved, and that you had the right geometry to hold everything in place. And because of this, and because Tom and Joan were thinking about RNA at the time, he also immediately noted that RNA enzymes involved in splicing might also use this mechanism for phosphoryl transfer. And a few years later, Tom and Joan together published this seminal paper where they published a general two metal ion mechanism for catalytic RNA and PNAS. But I think the other thing that really, I think, shows Tom's brilliance and how he was thinking about the problem, he was also thinking that this could be really quite an ancient mechanism, and that perhaps the RNA itself catalyzed the original polymerase or the original DNA synthesis reaction. And so maybe these two metals might actually be what was going to be the mechanism for the synthesis of DNA as well as for the cleavage of DNA. Well, as you all know, that actually is true, although at the time he proposed it, there was zero data to back that up. This mechanism has turned out to be incredibly general, and I swear I feel like it's following me around as I take on other projects. I don't have time to tell you about this today, but we're now working on a repair nuclease that we're able to observe using time-resolved crystallography, where we're literally watching in the bond getting, the phosphodiester getting cleaved by a two-metal ion mechanism that looks like the figure could be taken right out of this paper. These were things that Tom, we talked about jokingly when I was in the lab, but it was really quite thrilling to actually be able to see this, albeit on another enzyme. So, with this behind us, the goal was to try to get some primer template bound in the polymerase active site. A lot of people in Tom's lab were working on it, and we were all not being very successful. However, eventually, we did get a complex with DNA bound that I'll show you here. It's the first structure with DNA bound to polymerase. But it wasn't at all what we expected. And I vividly remember this day. I was working down in the basement of Klein Tower, where all the computers were, on trying to fit an electron density map with this density for this structure. And Tom came down, as he often did in the afternoon, and asked in his sort of teasing fashion, as he almost always did, to everybody, "Have you solved your structure yet?" And this time I said, yes, but it's not what you think. Because somehow the enzyme or the DNA had been remodeled. Instead of being a synthesis complex, it had turned into an editing complex. Instead of nicely going through the cleft and having its primer template here, we have the duplex DNA with a 3' single strand coming into the editing exonuclease site and going up in this direction towards the polymerase active site. I mean, you could not have come up with a substrate like this if you tried to design it. One of the real tricks, or one of the surprises, is that, and this turned out to be a general theme in polymerases, is that this was the first of many conformational changes that we would be finding in the enzymes that would really alter our thinking. So here's the apo-Klenow fragment, and pay attention to the thumb position here. And here's the picture of the Klenow fragment with DNA bound, and I omitted the DNA for clarity. There's a dramatic motion in this region, opening up a second cleft in which the DNA binds as it goes into the active site. So, although I was crushed at the time that I had failed yet again to get the complex of the synthesis complex with the polymerase, Tom noted immediately the implications of this complex that I didn't quite pick up on. And one of these implications was that his model had to turn around. The DNA absolutely had to turn around. So here we have a schematic of the DNA, or the single strand going into the exonuclease site and coming up to the polymerase site. And then in synthesis mode, it had to be coming in from this direction instead of coming through like this in the model. And this way, too, it fit all the available biochemistry thanks to this conformational change of the thumb region. Okay. So, although we never did get a complex of the Klenow fragment in the synthesis mode, before I left the lab, we were able to get a complex with a triphosphate bound in the active site, floating there nicely in the cleft. Although you don't see it, it's nicely coordinated by conserved residues, but no DNA anywhere in sight. And I was a little concerned this might be an artifact, but Tom scratched his head and said, no, it just needs maybe a few more pieces that we don't yet understand. And indeed, he was correct. So it was really looking back, I just feel very fortunate that I had an opportunity to work with Tom at that time and contribute to some of these early points in his central dogma timeline that is on his website. If you haven't seen his website, he has the central dogma and then his selected papers toward understanding the structural biology of the central dogma. And so we were just describing these first four from his greatest hits paper. But Tom went on then and worked with a number of DNA polymerases. The Taq DNA polymerase that's so important for biotechnology, bacteriophage, RB69, and Phi29 DNA polymerases, getting the DNA Pol3 holoenzyme much more recently. And I guess I should have the medically important HIV reverse transcriptase also on that list. The list would extend through many more slides. And I went on to Duke, ostensibly to be working with my colleague Paul Modridge on the proteins that are involved in DNA repair. And indeed, we are continuing to work in this area. And, but a little serendipity happened, and I was given a clone of a Pol1 enzyme by Strategene and asked if I might help them out on a problem. And this turned out to be an absolute dream come true for a structural biologist. Everything that we dreamed of doing with the Klenau fragment fell into place here. We have a polymerase that immediately crystallized in the synthesis form with duplex DNA coming into the active site. We had it diffracted to incredibly high resolution, 1.5 angstroms, which allowed us to really work out those details of mechanism. And then the reason I'm still standing here and talking about it today is another lovely twist. And that is that it retained catalytic activity in the crystal. So we were able to observe the precessive template-directed DNA synthesis in the crystal. Now, initially, this was a bit of again, serendipity. We took our crystals that were a DNA primer template, and we didn't quite understand what was going on because the template was going out of the active site and not through it, like the model would suggest. We thought, well, maybe we can figure out by soaking in some triphosphate and capturing a ternary complex before chemistry that way. But no, the DNA, we did this, and the nucleotide triphosphate was incorporated into the DNA, and the polymerase went and translocated by one in the crystal. Then in subsequent years, we were able to refine this, altering what we soaked in the crystals and extending the DNA out to blunt-ended. And this is a morph of several of the structures showing our current view of how a Pol1 polymerase works, where that triphosphate that was floating in space is delivered into the active site. And that you're getting this regulated transfer of the template strand from a pocket into the active site and chemistry happening. So we went on in this marvelous system to really be looking at all the mismatches bound in the active site just after chemistry. And we could really appreciate then how each one of them has their own story, but each one of them messes up that geometry so that you're not going to get subsequent synthesis off the end of the mismatch very readily, causing it to stall and then switch down to that exonuclease for those polymerases that have it. And one part of the story that I particularly liked, because it was really quite unexpected and something that, you know, I remember Tom and I chatting about at the time, which was, you know, how could the polymerase, there was chemical or biochemical data that suggested that polymerases could detect a mismatch even out of an active site and then switch it to be excised. And what we were able to do is to start with a complex, it's a schematic polymerase here, with a mismatch in the first position, and then by replication in the crystal, walk the mismatch from the first position through the polymerase until it left the other side. And it was only when you actually left the polymerase that you didn't see distortions in the DNA propagating back to the active site. And again, anything that messes up the geometry and the regulated transfer of the template would be likely to lead to stalling. And so what we found is that these long-range distortions in the DNA was really suggesting a short-term memory of mismatch incorporation that leads to stalling and then allows the polymerase, for those that have it, and this one does not, to switch from the synthesis mode to the editing, proofreading mode, and then back up again for synthesis, possibly through rotation of this thumb domain that we first saw when I was in Tom's lab. So, you know, pulling it all together, over the years, we identified a number of structural replication fidelity filters that contribute to the insertion and extension fidelity of polymerases. There are dramatic conformational changes. There is a regulated transfer, and effects of mismatches are seen from the pre-insertion site to the site of insertion, in the catalytic site, the post-insertion site, and back through the duplex region. There's so many parts of the structure that has to be beautifully aligned in order for accurate replication to take place. I think I will skip this and then leave you with the question of then, how do mutants happen? How do these polymerases make mistakes? They do on occasion, as we all know, and that's important for evolution. And so I'd like to end with a story that was one that Tom enjoyed and contributed to helping me get published, because at the time it was extremely controversial. So we were looking at mutagenic DNA replication with this system, and we chose to focus on the CA mismatch because that was a mismatch that I'll show you in a moment was able to potentially form a tautomeric species. And so in that animation, what you were seeing was the CA coming in, missing every error, missing one key fidelity filter after another, and coming in to the active site to be replicated. So the CA mismatch is interesting. In its normal tautomeric state, it would be unpaired, as you see here. But it had been observed to be ionized, and this was seen in crystal structures previously. And this is a wobble conformation, and with all of our work on mismatches, this would be readily picked up by the fidelity filters. However, it was noted very early on that the C could tautomerize to form a base pair almost identical to a Watson-Crick base pair in shape. And similarly, the A might be able to do this as well, forming this, rare tautomer that would be a cognate mimic. And this rare tautomer had been proposed but not previously observed, and many people did calculations to suggest that it was going to be too high energy to be at all relevant, if it ever existed at all. But the people who did these first proposals about the tautomeric form of bases and mimicking Watson-Crick base pairs were Watson and Crick themselves in one of their first papers. And this idea was then picked up on by Topol and Fresco, who advocated the rare tautomer hypothesis for spontaneous mutagenesis. But the work was I think, over the years called into question, and again, many a chemist calculated that it would be both too high energy to actually be relevant. However, so we set about trying to see if we could capture this CA mismatch and really observe its characteristics. So in our first experiments, we're just looking in the active site here, and we're looking at a superposition of the CA mismatch on top of a Watson-Crick TA base pair in the active site. And in green, we have the CA mismatch conformation, and in gray, we have the TA cognate conformation. And in this structure, basically, everything is working. The mismatch has been sort of captured. The helix is not closed where it needs to be. And most significantly, the nucleotide is not where it needs to be for chemistry. So the fidelity filter is working. However, we tried the experiment again with a mutagenic ion, manganese, that has been known to help stabilize that closed conformation. And there we got a radically different result. Now when you look at the BFCA and the TA, the structures superimpose perfectly. You could not tell them apart. And indeed, the CA mismatch has mimicked the shape of the cognate TA at the polymerase insertion site. And in this conformation, it would completely be able to evade any of the error detection mechanisms that the polymerase has set up. And just a quick look for the crystallographers in the audience. It is indeed at high resolution, so we can really see this conformation quite well in the active site. So what we have now seen is, for the first time, direct evidence for this rare tautomer. So we began to wonder, what is it, since it hasn't been seen before, what is it about the polymerase that might have contributed to this? And so we looked in the active site, and we saw that for all of the cognate base pairs in the closed form, that there was a water molecule interacting with the edges of the bases. And with that helps stabilize this and get everything in the right place for the two-metal ion mechanism of catalysis to work. And then when you had the CA in its more accurate conformation in the presence of manganese, this bond is not formed and things are not in the right position. However, when you get a little bit of help in closing down and dehydrating the site, the bond does form, possibly contributing to stabilizing the rare tautomer. There's a lot more I could tell you about this, but just to summarize, these CA mismatches can tautomerize and incorporate at low rates, consistent with the rare tautomer hypothesis of spontaneous mutagenesis. The polymerase, we think, contributes to this tautomeric stabilization in a number of different ways, and that the shift of a single proton can then have a very profound effect on fidelity. I'll stop with this and just share a few photos from my time as a postdoc at Yale. Basically, this was a pre-iPhone age, and I'm sure if I was a postdoc now, I would have many more photographs to choose among, but instead, there are really only a few in my collection. One that I love of Tom teaching the fine art of the barbecue to student John Arnez, and the more formal photograph captured by our wedding photographer with Tom and Joan sitting next to my mother at our wedding many years ago. And then this snapshot of one of many of these spontaneous lab gatherings at the ACS meeting shortly after Tom had won the Nobel. And I'd just like to close and say that Tom was a huge part of our lives when we were at Yale and afterwards, and that he trained a generation of scientists, and he will be always in our hearts. Thank you.